All right, welcome everyone to this week Autonomy Talks. This week is a pleasure to have Professor Simone Schürle-Finke from ETH Zurich. Something about her, she's an assistant professor at ETH Zurich, where she heads the Responsive Biomedical Systems Lab. Before that, she obtained her master's in industrial engineering at the Karlsruhe Institute of Technology. She then moved to Zurich, where she obtained her PhD in microrobotics, working in Brad Nelson's group. She then moved to Boston, and at MIT she worked on nanosensors for in vivo tumor profiling, as well as methods to enhance drug transport. Also in 2014, she co-founded an ETH spin-off called Magnetobotics, which offers electromagnetic control systems for wireless micromanipulation. In the past years, she received several awards, among which we note the Prix Zonta for Women in Science, the Branko Weiss Foundation Fellowships, and the Young Scientist Distinction by the World Economic Forum. With her group, she develops diagnostics and therapeutic systems at the nano and micro scale, and the aim of the group is to tackle a range of challenging problems in medicine, some of which we will see today. So we are very excited to hear what you're going to talk about, and go ahead, the stage is yours, Simone.
Thank you very much, Rede, for this kind introduction. It's my absolute pleasure to tell you today a little bit about the design and use of individual and swarms of microrobots for tissue probing and enhanced drug transport. I think since we're also a small group, feel free also to interrupt me if you have any questions. It's going to be very interdisciplinary. And I'm excited to get started. So I think we might all agree that medicine is becoming more personalized and targeted. And I believe that on this way, we actually need new tools to better stratify an individual patient's diagnostic state and help in therapy stratification, and also have new tools to be more targeted about the therapy that we want to bring to diseased tissue. And many people have been thinking about why not using nanorobots for that? And my background is in micro nanorobotics. I did my PhD at ETH Zurich, so my vision is to actually design and control tiny robots to detect and treat disease. So all we need to do is making large robots really small, right? That's what many people think of, and what you can find when you Google micro or nano robot. Then you find all these wonderful illustrations of, basically scaled down versions of large machines that are interacting on a single cell level, here with some red blood cells or some other clots in the blood vessels. But that's not how it looks like at the nanoscale. So what you see here are some real images, transmission electron micrographs of inorganic and organic micro nanostructures here actually, that actually can be used with some further engineering for biomedical purposes, and basically act as nanorobots for medicine. Because, and that we can understand this concept maybe by thinking of that for them also one of the fundamental principles in robotics also applies, which is that for a certain input signal, there's going to be a concrete processing rule that is applied and followed by a concrete output signal. And that is exactly kind of a fundamental rule that also applies to these systems. Why I think it is correct to call them a robot. And so how can we envision this now for such systems? So an input signal could be now a disease signal, such as an elevated pH, that could then result in an opening of a nanoshell and release of a drug, so for a therapeutic application. This input signal could also be an external physical cue that we can control, where a little bit more of the robotics and engineering is also coming in, that we're more familiar with, and it could be ultrasound, light, or magnetic fields. I've been working a lot with magnetic fields, so we could use, for example, alternating magnetic fields to open up again a nanoshell and release either again a drug, or potentially also a sensor that can then locally be released to bring us information about a disease state. And all these processing rules, the design of these systems, and the way of communicating with the system is informed by several disciplines coming together from chemistry, physics, biology, engineering, and medicine. And it's really truly a convergence of sciences. And this is kind of what we're also living in my lab, the Responsive Biomedical Systems Lab, where we basically design micro and nanoscale systems that respond to different environmental cues and physical cues. And today I want to focus this talk about around basically the use of magnetic fields for control of these robots in different applications in diagnostics, how we can use nanorobots to probe tumor activity locally to make better treatment decisions. how we can use microrobots to probe tumor mechanics in model systems. And lastly, when we then know more about the tumor tissue and can design appropriate therapies, how we can use synthetic and living microrobots to enhance ultimately the drug delivery and transport into diseased tumor tissue. So let's start with a nanosensor for disease diagnostics. So basically the concept is that we have a nanosystem that responds now to signals of the tumor, and we kind of get either a release of a marker or a shift in image contrast, which is kind of then the response or the way to communicate with these nanosystems. And now in a very concrete, let's go for a very concrete project I've been working on, where we developed a nanosensor for proteolytic activity probing of tumors. We'll come to that. So let's assume we have a patient who was diagnosed with colon cancer. Most commonly, biopsies are taken that are then analyzed by means of histology, or depending on where in the world and in which healthcare system, one is then also with genome sequencing. However, these techniques are basically analyzing that tumor tissue ex vivo, and we're missing important functional information of a tumor activity in vivo. We know, for example, that within the tumor, there are matrix metalloproteases, basically enzymes that reveal important, that orchestrate almost all hallmarks of cancer and reveal important information on a tumor's metastatic state. This is crucial information for a clinician in therapeutic decision making. And so basically these matrix metalloproteases, a type of enzyme, are now the disease cue that we want our system to respond to, so we can measure it. Now, how can we do that? Well, there exist certain peptide sequences, basically amino acid sequences, that can be coupled to a fluorescence marker, and they are specifically cleaved by certain proteases, by these enzymes. So basically, we have here not quite a lock key principle, but let's say a match of partners where we have a very high, there's a very high cleavage of these peptides when a certain target protease is present. Now, if we would now, though, want to detect this protease activity in the tumor tissue and intravenously inject such peptide sequences, we would encounter in the bloodstream not only the signal of the tumor, but actually also the signal of such proteases from many other organs, as these, you need to know that basically a certain specific protease upregulation in the tumor, which is a hallmark of disease, but the same protease is also important in many healthy processes in the human body. So basically this disease signal, which is because there's a lot going on in the tumor, but basically this is being washed out by a lot of signal of the other organs that also get shut into the blood. And so we have an issue with the signal to noise ratio to really detect the spatial local amplification of the certain enzyme activity. So we thought, can we not kind of design now basically a nanosystem as a delivery vehicle that can be intravenously injected and protect such sensors, such peptide substrates from the around floating proteases in the vessels, then accumulate at the tumor site and extravasate through the vessels, basically be small enough to leak through and accumulate in the tissue. And then once we're there, kind of release our sensing unit here, these peptide sequences, kind of like a Trojan horse now releasing the soldiers to measure. And we thought to do this activation scheme by basically incorporating into our protective nanoshell that is made of a liposome, a very common material used in biomedical applications and already approved, with magnetic nanoparticles that can absorb magnetic energy, and there are certain release mechanisms that can then open up the shell where the substrate can be released in the tissue. Now, how can we read out the activity of our sensing substrate with our fluorescence marker, for example, or any kind of marker that it's cleaved by a protease in the tissue? And so here we designed basically this the substrate with this marker now small enough that it can travel through the kidney into the urine, where we can read out if this substrate has been cleaved. We can read out the marker that is then released in the urine by urinary paper microfluidics. And so to tell you now a little bit more of how we did this. So this is basically the concept. You can see here now a transition electron micrograph of such a MAPS. We call it MAPS for magnetically actuated protease sensor. And so this here is this liposomal shell and these entrapped iron oxide nanoparticles. Here we basically, with dynamic light scattering, measured their size distribution here around 100 nanometer. So you can imagine this is now a liposomal shell. Liposomes are also a material how your cell membrane is made of. So it's very biocompatible and can be used as such delivery vehicles. And then we added here Triton X to basically destroy this shell and release our magnetic nanoparticles to show that we could entrap them. And indeed, by destroying the shell, we then see here the size distribution of the magnetic nanoparticles that were entrapped. So next, we wanted to see better how we can open that shell through dissipation, energy dissipation mechanisms. Here you see basically the specific loss power for these magnetic nanoparticles that we entrapped for different field amplitudes at an ideal frequency range for these particles for heat dissipation. And so we designed a coil that basically could entrap the size of a tumor for a mouse model to provide sufficient energy across this operating volume in terms of heat dissipation or dissipation to open this liposomal shell. So here you can see we basically have a release of payload upon magnetic activation. We first filled these liposomes with just with a dye. Maybe also for you as in context, this coil is designed here for a mouse scale, but the frequencies and field amplitudes we need are really scalable for human dimensions. So here, this is a system developed actually in Berlin by a company Nanofem. They do some magnetic hypothermia studies. And so a similar system could be used for exactly this mechanism we've been studying here in a mouse model. And we basically just showed that really it's not any background heating, and we have good control over stray heat, that when we have control liposomes without magnetic nanoparticles, and then we expose them to our magnetic actuation sequence, we do not see a release of our fluorescence payload. So in the beginning, we really just wanted to get this fundamental study. So we loaded it with a fluorescence that a molecule that has quenched fluorescence. So basically, when it gets released, we get a brightening of the signal. Now, when we expose it to our magnetic actuation cycles, we see this increase in fluorescence. We can even dose it. So with more cycles, we get more release. Or when we destroy our liposome, we basically can just get everything out. So this is kind of our threshold. So basically, now that we saw we could do this wireless activation mode of releasing payload from the system, we moved on and wanted to instead load it now with these peptide substrates that then target certain proteases. And so I don't want to dwell into much details, but there are lots of libraries for different MMPs or other enzymes that are important in certain pathologies, and they have different peptide sequences, and you see basically then you have very high cleavage, you have a red signal, and if it's a very low cleavage, then it's here in dark blue. And so you can basically find good matches here. If you want to, for example, look into MMP2, then this here is a very good sequence, as it has a very high affinity, and you don't get lots of cleavage by or noise by from also other proteases that would respond to this. So we designed in certain assays. I think I can jump you over, but basically showing that we got a very good readout for this certain peptide sequence for a certain target protease. And we went in vivo and wanted to see whether we can activate this nanorobot for release of local sensors also in a mouse model. So we have these tumor xenografts of a colon cancer cell line. We injected into two cohorts, basically our nanorobots, our MAP sensor, and basically without activation here, just as a baseline, have about the same urinary background signal. And then after 2 hours after, we locally applied our magnetic field actuation scheme, took again the urine, and then saw that we get this higher signal in their activated group, which is basically then showed that we could indeed open our shells in vivo. And next, we wanted to see whether we can now distinguish these enzymatic profiles in vivo and compare. So I mentioned in the beginning, it's important to get information about the metastatic potential of a tumor. So this is a cell line LS174T, which is very aggressive, and this HCT8 is not very aggressive. So LS174T is a higher likelihood for is basically metastasizing. And so we basically compared, we looked for now suitable peptide sequences that are very specific for this MMP2. So basically here, substrate 1 has a very high cleavage affinity. S3 is also strongly cleaved, whereas S2 is not cleaved, and exactly what we then saw. That's why we're talking here about profiles, is then for this aggressive cell line that is S1 and S3 is very high in signal, S2 not. And our non-aggressive cell line, HCT8, doesn't show such a profile, and basically no certain stronger upregulation for this MMP2. And we believe that, and you can adjust for different target proteases, enzymes you're interested, you can find different combinations. So here it's also a rich space for machine learning, trying to find best combinations, reducing background and so on. But, and there's a huge field around detecting and defining new protease substrates or peptide substrates for certain target proteases. And in the end, such tools, we believe, can help to inform results, can inform decisions in therapy. This work was then also published and received some news attention, and most importantly, there's now also a company. There was then also a patent. It's part of a patent, but many more studies are in that patent and concepts, and this is now actually already in clinical trials. Similar concepts based on these protease detection schemes. Now, I just told you about the specifics of basically this enzymatic activity in a tumor that can tell us about the metastatic potential. But so this is as a chemical cue, but there are actually also mechanical cues, and that deals with the stiffening of tissue around of the tumor matrix, basically, that then also is an indicator of metastatic potential and starting of invasion. And so this is also familiar with the concept of actually palpation at the doctor, where necrotic tissue and fibrotic tissue as part of this whole inflammatory process in cancer is basically hardened, which can be really macroscopically be experienced. However, it is quite different at the microscale, and also the stiffness and the shear moduli that are experienced and measured are quite actually across the scale, from when we look at bulk rheology measurements of collagen, which is one of the main constituents of the extracellular matrix in your tissue, or in humans in tissue. Versus here then for single fiber analysis. So basically this collagen is very fibrous, and if you pull on individual fibers, then shear moduli in the gigapascal range have been reported. Now there are different techniques to measure now the mechanical properties at the microscale with atomic force microscopy, where basically these tissues are scanned, or optical tweezers that can modulate the position of beads that can pull and twist and give an information about the mechanical properties of the interacting components, magnetic tweezers, also microrheology. And we thought to basically use now, again, magnetic microrobots for wireless 3D micromanipulation in these tissues to get an idea of the different stiffnesses in cancer tissue that could help us in some of the fundamental understanding of the disease progression. And so basically we wanted to fabricate, or we fabricated magnetic microagents at the scale, at the scale of single cells. So this works by, we synthesized these magnetic microrods by template-assisted electrodeposition, where basically you have a scaffold with holes and like cathode and anode. You can then basically, and having this in a solution with metal ions, you can then deposit the desired metal here, for example, cobalt-nickel combination on this on the substrate, and then release these particles. We got then basically these rods, because they grow. Then basically here this deposition grows over time. Then you can release them. We saw that we got quite a nice distribution of about 6 micrometer, or a length between between 4 and 10 micrometers, and a width of around 1 to 2 micrometers, and good magnetic response. We then functionalized them with NHS to bind with amines, and here you can see basically such an embedded micro rod in collagen that is labeled here with a fluorescent molecule, and here compared a human foreskin fibroblast, so kind of a similar scale and similar entanglement now in this matrix. And now how can we actuate these magnetic microrobots? So herefore we use a system that I was working on during my PhD, and you might have also heard earlier in the talk series from Brad Nelson's lab about a system composed of 8 electromagnets, where we make use basically of the linear superposition of the magnetic fields that are generated of each individual electromagnet, and can then basically apply desired fields and gradients in 3D, and therefore set certain torques and forces for a magnetic object in 3D. And we can, by a calibration I've been also working on during my PhD, basically connect through an inverse Jacobian as an actuation matrix the required currents to set a certain torque and to set a certain field and gradient, and therefore torque and force in a magnetic micro object. All right, so knowing now more about the tumor microenvironment, its enzymatic activity, but also potential mechanical characteristics, we now want to think of, and all this information can help us choose the right therapy or design appropriate therapy. We want to think about how we can bring that therapy to the location where it's needed, and enhance also drug delivery. So there is a rich portfolio of approaches in cancer therapy. Generally, if possible, the first mode of action is always surgery, usually combined with chemotherapy and radiation therapy. However, there are many new forms, including gene therapy, immunotherapy, which is very promising and up and rising, bacterial therapies and viral therapies, CRISPR-Cas9, as you also heard, and targeted nanomedicine. So all these basically need to, strategies need to get to the right location and only show their action at a disease site. So that's where in drug delivery and nanomedicine, actually we have a very rich history of different vehicles and ways of incorporating certain therapeutic agents. Again, you see these liposomal structures that I also showed you in the beginning. So this is really one of the oldest ones, was also approved by the Food and Drug Administration in 1995. There are also other polymeric shells and so on. And actually these liposomes are also used in the mRNA vaccines right now for COVID-19 vaccinations. So, but all these delivery vehicles actually have quite a challenge, and that is that, and this meta study from 2016 actually showed that only about 1% of the injected dose of nanomedicine from all publications reaches the tumor site. And all these studies on where we've been working on changing the material and the targeting strategy and the hydrodynamic diameter actually only marginally improved, maybe by 4 or 5-fold increase this accumulation at the tumor site. And when we think about this delivery problem kind of from an engineering perspective, it's pretty obvious that this is so hard, because we have a big pump, our heart, that pumps 5 to 7 liters per minute through the body. We have tubing with flow rates of 1 meter per second. We have encountered many mechanical filters. This dense tissue matrix I showed you. We have immune cells that take up these delivery agents and consider them as pathogenic foreign material. We have high pressure gradients, so there's a high interstitial fluid pressure in the tumor that actually works against any incoming molecular compounds. And so how can we actually work in this system? Could we not have something with like what you see here. This is actually a magnetic micro cylinder here of about 1 millimeter in length and 500 micrometer in diameter that is moved around in a silicon oil with that magnetic system I introduced to you in the beginning. So can we maybe apply a similar scheme for targeted drug delivery in vivo? Well, so can we basically power magnetic nanoshuttles in vivo and bring them by these magnetic forces to the tumor site? The problem is that the physics is not on our side with this. As you probably know, the magnetic field decays rapidly with distance. So the forces we can, or the fields and especially gradients we can generate at a distance are getting very small, and magnetic forces scale with the volume. So for a nanoshuttle, these forces get marginally small. And on top of that, actually also the fluidic drag forces are dominating as the surface to volume ratio is increasing. So how can we effectively still maybe use magnetic energy to power such shuttles? Well, if we look into how solutions in nature, what you see here is actually a macrophage, so one of these immune cells chasing a bacteria. And so bacteria have developed very smart ways of moving in liquids at a small scale and overcoming basically, or finding smart ways of moving at the low Reynolds number regime where these fluidic drag forces are dominating. So inspired by this intelligent propulsion scheme, many helical and micro and nanorobots have been actually proposed. So here just an overview of a couple that have been synthesized with different techniques, here with a glanced angle deposition, or actually also 3D printing in the end. So and they are all magnetic and can be basically actuated by rotating magnetic fields. And this is also a structure I've been then working on, the artificial Bacteria Flagella, or short ABF, that was developed in my PhD advisor's lab, which is was here basically fabricated by two-photon photolithography, so a 3D printing technique at a microscale that was then coated with magnetic material, basically nickel, and then titanium to protect the site, and then being actuated with this very same system by applying rotational magnetic fields. And then you can basically control such swimmers or artificial Bacteria Flagella ABF in host forms. And I've been wondering then whether this such an ABF could be used to basically enhance drug transport into diseased tissue when such an intravascular propeller is basically actuated and flowing against the bloodstream. And so we've first started to model this basically here in a two-fluid flow system, and looked into the local convection that is induced by such a micropropeller in a vessel. And what's quite exciting to see is, so at this scale, we're basically here in low Reynolds number regime, we have perfect laminar flow profile, undisturbed. But then you see here basically behind and before the position of the micro swimmer, you get basically a distortion of this laminar flow profile, and most importantly, the velocity components orthogonal to the channel. So basically out of the channel, outside of the channel extravasation, orthogonal to the fluid flow direction. And this is kind of here what you see also in these videos, how such a microswimmer is then in a to a fluid flow channel here, and you can see how it's then basically bringing up these nanoparticles to the upper part, and is also really creating here a wake around and uses local convection to push these particles away and to the side. And we then basically started to study this systematically in a model system where we created here. So this is a microfluidic device, contains here in the center collagen again that is mimicking the tumor matrix. Very simplified model, but it is the main constituent and can, depending on concentration, be very dense. And then here we basically have a microfluidic channel with these nanoparticles that are basically these dummy drug carriers that we want to bring effectively into the tumor into the tissue. And if they're only there, it's basically just a diffusion-based process And you see here basically this interface is created by so this is based on PDMS soft lithography. I can maybe in questions, if you have questions, we can discuss this later, but basically you can create also these these interfaces of collagen and the fluid by having these posts in between here, these polymer posts. And when the collagen as liquid is flowing in before it's polymerizing, basically here the surface tension effects keep it contained in the center area, and we can create these interfaces of fluid exchange into this tissue. And this is how such a device then looks like, and that is then put under the microscope. Here again in the vessel, in such a model, what we would expect is for an incoming nanoparticle stream, and with such a micropropeller sitting there, how we can actually then increase here the transport that is otherwise diffusion limited into the tissue model. And we studied then also basically again how this distribution would look like for these different outlet areas around this micropropeller, and we expected basically such kind of a profile, and based on these simulations. And this is also what we basically then saw by analyzing now these outlet regions at the side of the propeller before and after, that we get an increase here during actuation by of the nanoparticle intensity if we have such an actuated system compared to controls where no ABF was present. What we also saw is that we could actually increase also the penetration depth of nanoparticles here by basically showing how we could we measured basically at the beginning and end how the intensity profile away from the distance from the vessel. And so this was an encouraging result of whether we could use maybe such intravascular propellers to steer up nanomedicine. But what we also saw, and this data I actually didn't show, but it is very dependent on the distance of the propeller to the boundary, or the ratio of the size of the propeller to the size of the vessel. And you can also imagine that this might work well then in smaller vessels, but overall it would be great to have actually more of such propellers and have them very densely in the vessel, as the strongest effects are the closer we are also here to the wall, where we basically have no where we have no velocity. And but it's getting very tricky to control several of these micro swimmers because they're all magnetic. So we have magnetic aggregation, and this can also actually form dangerous clots in that sense. So how could we control whole swarms? And it turns out that, again, nature might have already a solution for us. So what you see here are actually real bacteria that contain, though, iron oxide nanoparticles, basically mainly magnetite, they biomineralize by taking up iron from their environment. And these bacteria do this to navigate with the Earth magnetic field in aquatic environments. So we can basically now, by exposing them to an external magnetic field and letting them just swim and propel by themselves, we can give them a direction. But what's even more exciting is that by applying rotational magnetic fields and mixing them here with, again, such fluorescently labeled nanoparticles, we can generate large volumetric flows that can move these nanoparticles with them, and we thought potentially also drive them into tissue matrices. So using this concept and maybe just to also make this very clear, so we could either apply directing magnetic fields and use their own propulsion. If we apply rotational magnetic fields, they swim in plane, they basically would swim in circles. But if we now step out of plane, and have this friction and interaction with the surface, they actually start tumbling and moving forward. And because if they're very dense, they basically experience this resistance from neighbors. And so we start generating this whole flow once we have one layer that has this boundary interaction. And so we started to track their velocity and motion, and also this concept is well known in the field of ferrofluids. So usually ferrofluids are colloidal suspensions of magnetic particles, magnetic nanoparticles, usually in the subdomain size. But here now we basically have ferrohydrodynamics of a living ferrofluid, and so we see though that this behavior is quite similar. And I'm just checking the time to that this behavior is quite similar to these ferrofluids that have been studied before. And we basically have a peak frequency. And after that, it's basically where the fluidic drag forces are dominating. So this peak frequency or step out frequency can be pushed to higher frequencies and thus then also higher velocities by increasing the external field strength, as we're then increasing again the magnetic force we're applying. And so we studied then this the scheme in the same device. Basically now we just looked into the integrated increase here of intensity of these nanoparticles in this tissue area, and we could see that when we actuated these bacteria over time, we could increase also the amount of particles that we got into the tissue, especially also the change per minute. So the velocity was much higher compared to diffusion-based approaches. And which was also really exciting is that we could switch on basically when we come here into saturation of diffusion, we can induce convection and start again this process of transport. This was also captured as nanoparticles take a fantastic magnetic voyage with these bacteria. When there was an animation wrong, but basically what we then also now started to study in my lab was going deeper into the actual ferrohydrodynamics of these living ferrofluids compared to synthetic ferrofluids. And so what we basically did was comparing the fluid flow that we could generate between these magnetotactic bacteria and these classical ferrofluids that are composed of iron oxide nanoparticles, and they would start to chain and cluster, and basically, so we recorded their typical velocity profiles under certain external magnetic fields, and what we found is that basically, if we normalize this as a figure of merit to the magnetic material they bring in, because of course these bacteria have a much larger hydrodynamic diameter, they also don't aggregate. And basically they achieve much higher velocity per magnetic material for a certain number of actuators. And what is also quite exciting to see is that their flow fields are much more homogeneous, and that is because they don't start to cluster. So we basically get this here, the deviation from the mean velocity is almost zero around, whereas we have large variations for these synthetic ferrofluids because they start clustering. And that is also obvious in these videos where you will see on the left for the magnetotactic bacteria, it stays quite the same, but on the right, we have more clusters. And we also confirmed this basically by measuring their size before and after actuation, where we got a significant increase in size for these iron oxide nanoparticles. Now, you might think, okay, it's great that these bacteria are such good flow actuators because of their hydrodynamic properties, but you don't want to use them in vivo. Well, we actually do. We believe that bacteria can be used for cancer therapy, and this idea is actually not new. So their bacterial cancer therapy is an old concept. There was an immuno pioneer, actually a bone surgeon, William Coley, who discovered in cancer patients a regression in tumors when they also had a bacterial infection. And we kind of think about bacteria as a living microrobot, in a sense that they can, because what was also seen is that they can home to tumors. It's an environment they like to grow. It is low in oxygen. They can provoke an immune response and have onboard drug production. And so overall, now with tools in synthetic biology, this is a renaissance of an old concept we are working on. But I think I'm unfortunately at the end, so I have to go really quickly through these last slides. I'm very sorry about that. I was talking longer before than expected. So there is basically, for you to know, it's an exciting field of these genetically programmable therapies. And we want to add an additional level of control through external control here with magnetic fields. We've been empowering bacteria with magnetism in the sense that we work with these magnetic bacteria. We give them certain drug payloads with them. We study how they also interact with immune cells, and then basically created physiological barriers and studied how we can help them to overcome these physiological barriers. So here with their drug cargo, before we showed they can help the drug cargo to get into tumor tissue, but now we want to actually get the bacteria with their attached drug cargo and use them as a living therapeutic vector. And we could show that with this actuation scheme, we could actually better cross cell monolayers here for models of the vascular endothelial monolayer that you encounter in vessels. We started also to model these mechanisms. So unfortunately, there's no time now to go through this, but basically they this rotational fields can help them to explore surfaces and find gaps, because these endothelial cells, they actually open and close, so they form gaps where you can go through. But through this mechanism of surface exploration and guided actuation along these cell gaps, we can actually better find basically gaps to come through and penetrate. We also then studied how we could detect them using rotating magnetic fields with an inductive detection scheme. So we can also follow them in vivo. We can use it for closed loop control and real-time monitoring. And we've also seen that we can basically enhance their penetration then in the end in these tumors for theories that I showed before, these models of a tumor, basically bring their cargo there. Here you can see this very nicely, how with this actuation control, we get these bacteria here labeled in red deep into the tumor, versus for the control, not so much is going on, and they actually even grow and amplify over time in the tumor. We design systems to spatially control then also the area of action by superimposing rotational magnetic fields with static gating fields, to basically not expose the whole body to these rotational magnetic fields and be more selective. These are also new systems we're now designing for mouse scale system. And lastly, we've been using these bacteria also to compete for nutrients here for iron. I told you they need iron to synthesize the iron oxide nanoparticles. This is a scheme in therapy. We've been basically showing that these bacteria alone can also induce, and this is shown here, cell or reduce cell viability because they starve cancer cells for iron. And with that, I'm really now at the end, and I hope I could give you an idea of different modes of action in diagnostics and therapy with synthetic and living individual and swarms of microrobots. And I'm open for any questions, and thank you for your attention. And most importantly, I need to thank my team, because they did all the work. And also the beautiful science illustrations that you've seen are from Sonia Monti, who's actually also in the audience. Thank you, Sonia. And of course, big thanks to all collaborators and funding.
Thank you, Simone, for the great talk. So let's open the questions from the audience. Do you have any questions?
Thank you, Simone. It's a very fascinating material. It's amazing. I had no idea that you guys went so far. But now, okay, let's start with the meta question. So, as I was following you, like all the things that you were saying, right? So this is amazing, right? All the potential for everything that you can do with this kind of technologies. But then you think of the reaction that people have been having to the vaccine for the COVID-19, right? And people, some people, especially here in America, they don't want to get the vaccine because they're afraid of, Bill Gates is injecting them with a chip, right? So now what if you tell them, I'm going to inject you with a whole bunch of nanorobots? And have you had an idea of what would be people's reaction to these kind of things?
Yeah, I think that's a great question. It depends. So some are excited about it, and it always depends on how you phrase it, right? Some call this nanorobot. You could also call it a molecular system. I think it also depends on basically how you sell it, how to whom you're talking, how you should sell it. And definitely there have been people be concerned about the idea of having basically microrobots or nanorobots in the body. But if you think of what they actually do and what they're composed of, how they also our control of getting rid of them or control. So we've been also thinking about ways of destroying them with magnetic fields, of, we control their site of action. There's of course also clearance, like there's a lot around it that makes this, and that makes this safe, in my opinion. And and the other point is, for many of the diseases we're targeting, it is so it's so terrible. Like
okay.
and I think
it's which is different from a vaccine that you get when you're healthy, right? Okay. So another thing I was thinking of is I think that most of the techniques that you use are mostly open loop, right? Or is there any way that you can enable some kind of feedback, right? Because I guess that when you collect the the little robots, right, and in the urines, and then you look at how many of them you have open and things like that, that's kind of like allows you to do some form of integral feedback, right? In a sense, right? But are there other mechanisms? For example, how do you know where in the body all of these nanoparticles, nanorobots are? How do they distribute?
Great question. So actually, also a PhD colleague during my time as a PhD student in Brad's lab, there was a colleague working on a combination of magnetic particle imaging, so not MRI, MPI, you directly get a magnetic response and no tissue contrast, so no hydrogen interaction. So really just MPI in sequencing of control, basically combining control and imaging. This is one idea. We've been now working on this inductive detection scheme also. So basically you could have a sense and compensation coil. You could basically, give a short, even now with RMF, we did this also, right? Getting basically the inductive signal from the bacteria with their magnetic components through inductive detection, which would also be lower cost compared to an MRI. I think this is an interesting direction to actually look at response.
Okay. Thank you, Simone. I don't know if somebody else has other questions. I don't want to talk all the time.
Are there any other questions? I have one related to the last one Emilio asked, and it's about, I think it was slide 63 or 64. So related to the closed loop. I wonder if maybe at some point, you need to switch between these in-plane and out-plane controls, right? So either directly steering the robots or causing these flows that make them move. And my question was, so in view of the closed loop, or in view of knowing where your robots are, how reactive are these kind of systems? So if you want to switch the direction, or if you want to switch the kind of control you are applying, I guess you need to be very quick, because maybe you are missing the target if you don't.
Actually, under control of the magnetic field, it's also because we have basically no inertia. It's like immediate switches. You could also see in the video, right? This is this was not but it is immediate response. What we do actually also, because you can imagine there are so for in vivo, we actually did first in vivo trials, but very preliminary data, so I didn't show that. But we were basically changing the direction of the magnetic field in a sphere, like the rotating vector would just go around a sphere, because you in one direction you have many you want to create orthogonal interfaces, and this is a complex environment. So basically we just sweep through, if that answers it. But basically they you don't need very strong fields. These magnetic bacteria respond to the Earth magnetic field, which is around 50 microtesla, and we just apply 5, 10, 20 millitesla and superimpose that, and then it's immediate response.
All right. So meaning that if in case you would have a perfect state estimation, the actuators would be able to actually use this information. That very cool. Very cool. Are there any other questions? I guess maybe a question that arises. So we are using this group to think about adopting new technologies in terms of autonomous vehicles and all the testing that is required, the regulations. I guess you would have to go through the medical ones, and on top of that the robotics ones. So some words about that. Is it difficult to get these technologies approved?
Yeah, I mean, so with these magnetic bacteria, that might be really tricky. So that's why we're working already also with other bacteria that are right now in clinical trials and where we put magnetic materials on. So we're trying to build up and piggyback on things that are in trials at least. So, and for the magnetic control schemes, I'm actually not too worried. Out here, we're building up on other things, right? Magnetic catheter control. So it's the same input fields as MRI. I mean, there's a lot of things where we can basically say, okay, this has been shown to be safe. And so where I think the translational barrier is lower. And also we see more and more research really on, especially the living therapeutics is a big thing also for the microbiome, for the whole digestive tract. Now with bacteria, recently also a study with magnetized bacteria. A lot of people still rely on these, putting a permanent magnet somewhere on your abdomen and use gradient magnetic fields, and that just doesn't scale. So I truly believe that these concepts of rotational magnetic control and using these flow fields can be a good solution to that. I mean, yeah.
Very cool. Thank you. If there are no other questions, maybe let's wait 5 seconds for somebody that has a last one.
Also, I know it was very fast and a lot of new stuff, so
exactly,
don't hesitate. Or also afterwards, if someone wants to know more about something.
Yeah, that's very orthogonal to what we are used to learn. But
I should have maybe, yeah.
Okay. No, in the sense that there is huge potential, right? Cool things happen at the intersections usually, right? So
maybe it inspired something.
Actually, something that I would like to say is that as I was, following your talk and looking at your slides, I was very jealous about your illustrations.
See, I mean, I told you the secret. She's told you the secret, yeah.
Very impressed. It's like good to know. I think that we can rely on her if she has some time. That's great.
Super.
All right. Thank you very much, Simone, for the great talk, and good luck for all the next projects. As I said, if somebody has further questions, feel free to reach out to Simone. And yes, thank you again very much. And see you all next week for the next Autonomy Talk.
Thank you. Bye.
Bye. Thank you, Simone.